In this video, I will discuss the concepts and enzymes involved in a prokaryotic replication fork. As a reminder, a prokaryote is a single-celled organism that does not contain a nucleus. DNA is the genetic material of organisms. DNA is made up of nucleotides, which are composed of a sugar, phosphate, and a base, that are held together by phosphodiester bonds. DNA replication is one of the three major information pathways in the central dogma of genetics. It is the process of copying a double-stranded DNA molecule to produce two identical daughter DNA molecules. It is also important to note that DNA replication is semi-conservative, which means it uses an old strand of DNA as a template to direct synthesis of a new complementary strand, and the resulting double-stranded DNA is half old and half new. There are several models of semi-conservative replication, but I will be discussing the theta replication model in this video as it is the most common mode of replication for prokaryotes, such as E. coli. Prokaryotes have circular DNA and only one replication bubble during replication, which can be seen in this diagram of theta replication. This image shows some general terms surrounding prokaryotic DNA replication within the theta model. The origin of replication is the location where replication begins. Prokaryotes only have one of these regions, whereas eukaryotes can have many. Also notice how the 5' and 3' ends are labeled on this diagram. DNA replication can occur only from the 5 to 3' ends, so identifying these regions is crucial for understanding the process of replication. In order to define specific concepts and enzymes involved, we'll take a closer look at just the replication fork. Looking at this image, we can identify the leading strand, which is replicated continuously, and the lagging strand, which is replicated discontinuously. Discontinuous replication, as occurs on the lagging strand, results in small pieces of DNA called Okazaki fragments, as shown in this picture. Now back to our previous diagram of the replication fork. The molecule at the very top here is gyrase, which is an enzyme that relieves tension due to supercoils caused by strand separation. Next, we have helicase and primase. Helicase unwinds the DNA double helix while primase lays down RNA primer since DNA polymerase requires a free 3' OH end. This molecule represents a single-strand binding protein, which binds to the strands of DNA to keep them separated during replication. These two molecules represent DNA polymerase III, which is an enzyme that attaches to the primer and adds nucleotides. This is the main polymerase in prokaryotic DNA replication. Finally, we have DNA polymerase I and ligase. DNA polymerase I removes the RNA primer and replaces it with DNA. Ligase completes replication by sealing the new strand and repairing the remaining nicks in the DNA.